the last video, we have discussed about the fertilization events which takes place in case of plants. And now in today's video, we will discuss about the post-fertilization events. Before that, let us recall what had happened in the fertilization event. During fertilization event, we have seen that the stigma part of the pistil, it receives the male gamete. It will germinate here, and then the formation of the pollen tube takes place. Through the formation of the pollen tube, the two male gamete along with the tube nuclei, they enters into this endosperm through the micropylar end, and after going inside it, one of the male gamete fuses with the egg and it leads to the formation of zygote, whereas the second male gamete moves towards the central part and it fuses with the polar nuclei, which leads to the formation of endosperm, all the primary endosperm nucleus. All these events have taken place during the process of fertilization, and as a result of it, we have seen that inside this egg apparatus here, due to the fusion of the first male gamete with the egg, the formation of the zygote takes place, which is diploid in nature. And the second male gamete, it enters into the central region, and here it fuses with the polar nuclei, and here, because of the fusion with it, it leads to the formation of primary endosperm nucleus, which further develops to and lead to the formation of primary endosperm cell, which is triploid in nature, and this is the nutritive tissue that has been formed as a result of fertilization. Now, today we will discuss the post-fertilization events. After the process of fertilization, which is also known as double fertilization in case of angiospermic plants, some changes takes place in the flowers, which are the part of the post-fertilization event. Now, what happens during which? We know that after the process of fertilization, first of all, we will write about the post-fertilization changes. The different post-fertilization changes which takes place in the flower are the petals, stamen, and sometimes the calyx shed off. As the petal has performed its role to attack the pollinating agent, stamen, its role was to perform the male gamete, and this male gamete has come here and it has performed its function of the fertilization. Now it is of no use, so both of them, they will shed off. Now, in some of the cases, the sepal may shed off, whereas in other cases, sometimes the calyx, it may not shed off, it will remain with the floral parts. The next event here is the role of the stigma has been completed, whose role is to receive the pollen grain, so it has performed its function. So the stigma and style will degenerate. The stigma has performed its function to receive the pollen grain. The style, it has helped in the formation of the pollen tube. Its role is being over. Hence, both of them, they will shed off. So, they will start degenerating. They will start degenerating. And only which part is left? This part will remain on the plant body, which consists of, this is the ovary will remain there, And we know that inside the ovary, this is the integuments. Two integument layers are there, and this part is the ovule. Now, after these changes, what happens that this ovary will develop and it will lead to the formation of fruit. We know that the ripened ovary is known as fruit. So this ovary will develop into fruit. Ovary develops into fruit. The next one is that this ovule, which is normally single in number, and in some cases the number of the ovule may be many. So this ovule, this is the ovule part, it will develop into the seed. So ovule develops into seed. And what happens here? This is the integument part. This integument will lead to the formation of seed coat. So integument leads to the formation of seed coat. Now, the most important part of the post-fertilization event is, we have seen that as a result of the fertilization, the formation of the zygote and the formation of primary endosperm nucleus have taken place. So now after this, what happened? This zygote, it will develop into the future embryo, whereas this primary endosperm cell, it will result into the formation of endosperm. And we know that the endosperm is the nutritive tissue. So inside this ovule, the development of zygote and endosperm formation takes place. Now the question is whether the zygote is going to be formed first, or it is going to be developed first, or the endosperm development will take place first. Here the answer is, first of all, the formation and the development of the endosperm takes place, and that is being followed by the development of the zygote into the embryo. In the meantime, we have to keep in mind that around this embryo sac, it is being surrounded by the layer of nucellus cells. It is being surrounded by the layer of nucellus cells. So this is the nucellus cells. Now what happened? During the development of the seed, most of the time those nucellus gets consumed or used up, whereas in some of the case, even after the formation of the seed, sometimes some nucellus persist. That means even after the formation of the seed, some of the nucellus cells, they persist, and this type of nucellus which persists even after the formation of the seed is known as perispermic seed. So that type of seed having the persistent nucellus are known as perispermic seeds. So the perispermic seeds are the seeds in which even after the development of the seed, some amount of the nucellus remains there. The example of this one is beetroot, black pepper. So these things we have discussed here. Now coming to the main point, we have discussed that after all these events, the main focus is the formation and the development of the endosperm as well as the formation and the development of the zygote into the embryo. First of all, we will discuss about the formation and the development of the endosperm. Endosperm and its development. We know that the endosperm, it is the nutritive tissue. Endosperm is the nutritive tissue and its role is to provide the nourishment to the growing embryo. So it is also acting as the reserve food. It also acts as reserve food because it contains the nutritive materials which help in the development of the zygote. Now, on the basis of the development, the endosperm can be classified into three types. Endosperm is divided into three main types. These are free nucleated endosperm. The second type is the cellular endosperm, and the third type is the type that we have to discuss is halobial type of endosperm. One by one, we will discuss about these three types of the endosperm. The first type of the endosperm is the free nucleated endosperm. Here, actually what happened, we know that this is the primary endosperm nucleus. This is the primary endosperm nucleus and having 3n number of chromosomes. It is the diploid condition. It results into the formation of primary endosperm cell, and after the formation of this primary endosperm cell, what happened? It will undergo continuous nuclear division. It will undergo continuous nuclear division. That means it will undergo continuous karyokinesis without undergoing cytokinesis. Nuclear division is going to be taken place, but no cytokinesis is going to be taking place. As a result of this, what happened? After some time, a large endosperm structure is formed, inside which multi-nucleated conditions are there, and all of them they are triploid in nature. So in this case, what happened? A single endosperm, it will develop continuously and it will divide by the nuclear division without following the cytokinesis. Hence, as a result of this, a cell structure is formed, the endosperm is formed, which contains many nucleus. So this is the free nucleated endosperm condition. The example is the case that we have seen in case of coconut. You may have seen in case of coconut that the water part of coconut, that we have seen the water part of the tender coconut, that is better to say tender coconut, which we have observed in the case of the coconut, that is the formed because of the result of the free nucleated endosperm. The second type of the endosperm is the case where here also this primary endosperm cell is present, which is 3n in number, that is diploid in nature. This endospermic cell will undergo cell division and it will lead to the formation of two cell stage, and all of them they are having 3n number of chromosomes. That means karyokinesis is followed by cytokinesis. So here also cell division takes place, but here karyokinesis is being followed by cytokinesis. After this cell structure further divides and it will lead to the formation of 4 cell stages, and all of them they are having 3n number of chromosomes. So in this case, as a result of this, after some time cluster of cells are formed because of karyokinesis followed by cytokinesis, and after that the formation of the cell wall takes place, and that's why this type of the endosperm is known as cellular endosperm. The example of this one is the example of this one is also the coconut, but the white part of coconut that we have seen. Actually, what happened in this case? In case of coconut, we have seen that there the free nucleated endosperm as well as cellular endosperm takes place. How this is possible? Let us discuss. Actually, what happened in case of this coconut, if this is the endospermic cell or the nucleus, this endospermic nucleus first of all undergo free nucleate division, and it will lead to the formation of the condition where it will lead to the formation of multinucleated condition. That means first of all it will lead to the formation of free nucleated endosperm. In the beginning, karyokinesis is happening, but it is not going to be followed by cytokinesis. As a result of this, in the same cellular structure, the formation of many nucleated condition is formed. But after some time, what happened? The nucleus, these nuclear cells which are present on the peripheral region, these nucleated structure, they will undergo karyokinesis followed by cytokinesis. So the outer structure will result into the formation of cellular nucleated endosperm, and because of this we have seen in case of the coconut that inside it the watery part is there, which consists of the free nucleated endosperm, and to the in the peripheral region we have seen the cellular nucleated endosperm. So this is the structure how the formation of the coconut has taken place, and that is the endosperm of the coconut. Now coming to the third type, that is known as the halluvial type of endosperm, that is generally found in case of monocots. It normally is found in case of the monocots plants, and here actually what happens in this case, first of all, if this is the endosperm cell having 3 number of chromosomes, first of all it will undergo the cellular endospermic nucleus. It undergoes karyokinesis followed by cytokinesis. As a result of this, we can see the structures are formed where the number of cells which are formed, all of them they are having 3n number of chromosomes. But after this, what happened? The cells which are present at the lower part or the micropylar region, these cells they will start dividing, and these will here the free nucleated type of endosperm formation takes place. That means karyokinesis happens, but it is not being followed by the cytokinesis. Hence we can say halluvial type of endosperm, it is the intermediate of free nucleated and cellular endosperm. So, on the basis of the development, the endosperms are of three types: free nucleated endosperm, cellular endosperm, and the halluvial type of endosperm. We have already discussed that the endosperm, it is the nutritive tissue and its role is to provide the nourishment to the growing embryo. This is the most important function of the endosperm. Now, what happens when this formation of the zygote or the embryo takes place? In most of the cases, the endosperm get consumed during the development of embryo, whereas in some cases some amount of endosperm remains even after the formation of embryo. So on that basis, the endosperms can be classified into two types. Those endosperms which get consumed during the development of the embryo, these type of endosperms are known as the seeds which contains these type of endosperm, they are known as non-endospermic seeds. That type of seeds are known as non-endospermic seeds, where or we can say or non-albuminous types of seeds, whereas those seeds which contain some endosperm even after the formation of the embryo, that type of seeds are known as the endospermic seeds, or they are also known as albuminous type of seeds. The example of the non-endospermic types of seeds are, we can say the case of pea, bean, etc. Whereas the example of this endospermic type of seeds are, castor, coconut, etc. So in this video we have discussed about the formation and the development of the endosperms and how they are being classified. In the next video we will discuss how the development of the zygote into the embryo takes place. Thank you.